DNA is used to make proteins. That's its main job. DNA is used to make a copy of mRNA, where DNA is GCANT and mRNA is GCANU. This is called transcription, and it happens inside the nucleus. Then a ribosome will read the mRNA, which is made up of GCANU, to generate a polypeptide, which is a protein, out into the cytoplasm. This is called translation.
Extraction of DNA. In order to extract DNA, you first need to add salt and soap to cells, for example, the cells of a strawberry. In order to extract DNA, you first need to add a mixture of dish soap and salt to your mashed strawberries. This aids in extracting DNA because the dish soap dissolves the phospholipid cell membranes of the strawberry cells, and the salt breaks down the protein around the nucleus, allowing the DNA to be released for extraction. The next step of extraction is adding cold ethanol to the mixture of strawberry soap and salt, which allows the DNA to come together, making it visible and solid enough to extract.
Ligase is what holds the strands of DNA. Primase is the complementary strand of DNA to a single strand of DNA piece. Helicase separates the double-stranded DNA. Primer is a short strand of DNA that provides the starting point for the DNA replication. Polymerase is what makes the other side of the DNA strand and completes them to make two DNA strands. The lagging strand is the bottom half of the DNA that gets separated, and the replication does not happen constantly on it, leading to okazaki. The leading strand is the top part of the DNA strand that has the replication happen constantly on it.
Polymerase chain reaction, otherwise known as PCR, is a laboratory technique used to amplify DNA sequences. The temperature of the sample is repeatedly raised and lowered to help a DNA replication enzyme copy the target DNA sequence. The technique can produce a few billion copies of the target sequence in just a few hours. The three main steps in a polymerase chain reaction are denaturation of the template into single strands, annealing of primers to each original strand for new strand synthesis, and extension of the new DNA strands from the primers.